Hi, I'm Yihong Ong from University of Pennsylvania. Today my talk will be focusing on numerical investigation using Monte Carlo simulation to estimate the depth of tissue probed with different source and detector separation. Photosensitizer concentration and tissue optical properties are two very important parameters that are commonly measured during PDT dosimetry. Photosensitizer emits fluorescence light when excited by the treatment laser, and this fluorescence light allows quantitative measurements of the spatial distribution and temporal changes of its concentration in vivo. The second parameter, tissue optical properties, helps improve light dosimetry and guide light delivery. Information about the spatial heterogeneity as well as the changes of tissue optical properties during PDT are important and need to be accounted for to improve the accuracy of photosensitizer concentration quantification. Here at Penn, we have developed a broadband diffuse reflectance and fluorescence spectroscopy system with a handheld contact probe design that allows for the measurements of these two parameters during clinical PDT. On the left is the schematic drawing of our dual-mode spectroscopy system. Basically, it consists of two light sources, a halogen lamp and a 405 nanometer blue laser, a fiber bundle of two source and 9 detector fibers, a monochromator, and a liquid-cooled CCD camera. The fiber bundle is built into a handheld contact probe design, as shown in the right picture, with a trigger button to allow for the physician to control and take data from various tissue sites during clinical PDT. This is the zoom-in view of the tip of the contact probe. Two source and 9 detector fibers are arranged in a single line. Each fiber has a core diameter of 400 micrometers and a numerical aperture of 0.22, which is equivalent to a half cone angle of 12.7 degrees. Source fiber 1 is connected to the halogen lamp, and source fiber 2 is connected to the 405 nanometer laser. The 9 detectors fibers are spaced non-uniformly. The source detector separation for this design ranges from 6 mm to 1 cm. For broadband diffuse reflectance spectroscopy, in order to solve for the two important tissue optical properties, which are the absorption and reduced scattering coefficients, we need to have at least two input variables. One of these variables comes from the use of white light spectrum that has different wavelengths, and the second variable comes from the source and detector separations that create different light path lengths. Although different pair of source detector fibers allowed us to vary light path length, the volume or the depth of tissue sampled by each pair of fiber could be very different. The knowledge of how light propagates in tissue is helpful and necessary for us to interpret our measurement results. For example, from which depths the measured fluorescence is coming from, and is the sampling depth changing with the choice of source detector separation. To understand and to help answer all these questions, a numerical investigation using Monte Carlo simulation was performed to specifically look into the effect of source detector separation and tissue optical properties on light propagation in tissue. We also attempted to estimate for the depths of tissue probed by each source detector pair for both diffuse reflectance and fluorescence spectroscopy. For the fluorescence study, we simulate the excitation and emission of fluorescence signal for photofluorescence sensitizer, which typically involves excitation in the blue and detection in the red region of the visible spectrum. This is the setup geometry for the Monte Carlo simulation. Generally, a semi-infinite tissue model with a single air tissue boundary was used. The refractive index of air is 1, and the reflective index of tissue is set at 1.37. Uniform tissue optical properties were used, and the concentration of fluorophore was assumed to be homogeneous within the tissue. On the boundary between the air and tissue interface, we introduced a source fiber and a detector fiber. The core diameter of this fiber was set to be 400 micrometers, and the numerical aperture was set at 0.22, similar to those fibers on our contact probe. The distance between these two fibers, ρ, range from 1 mm to 1 cm in this simulation. This slide shows the flowchart of events in the simulation. For every source detector separation, at least 1 million photons were generated. For each photon launched in the simulation for diffuse reflectance, it propagates and it deposits a fraction of its weight every step until it reaches the detector, or when it is fully absorbed in the tissue, or when it escapes into the air through the boundary. Then a new photon is generated, and the process continues until every photon is simulated through the same events as depicted in the top half of the flow diagram above the red dash box. For fluorescence simulation, it involves two photons, the excitation photon from the source and the fluorescent photon generated within the tissue. These photons propagate with two different optical properties. For every step taken by the excitation photon, a new fluorescence photon is generated. This fluorescence photon will propagate and go through the events as shown in the red box. To be more efficient in simulating these fluorescence events, and for the ease of data interpretation, only photons that reach to the detector fiber within the acceptance cone will be recorded and studied. For fluorescence simulation, we specifically look at the excitation and emission signal of photophrene, which is a type of photosensitizer that we are working with both in preclinical mouse studies and in clinical trial. Photophrene has strong absorption in blue region of the visible spectrum. And in our spectroscopy system, a 405 nanometer laser is used as the excitation source to generate fluorescence efficiently. Photophrene emits fluorescence in the red region, between 600 to 700 nanometer. To mimic the generation and detection of fluorescence light using our contact probe setup, we simulate the excitation light at 405 nanometer as depicted by the blue shaded area on the plot. And for the fluorescence light, we run the simulation at 630 nanometer, which is at the most prominent peak of the entire fluorescence spectrum. The main inputs of the simulations are the absorption coefficient and reduced scattering coefficient. The selection of this number was to mimic the optical properties of human tissue as closely as possible. Hemoglobin is the main light absorber in human tissue, and in our simulation, we select the optical property at both the excitation and emission wavelengths based on the molar extinction coefficient of hemoglobin shown on the left. The absorption of hemoglobin changes quite a bit with its state of oxygenation. A fully oxygenated blood has much lower light absorption at the red region compared to fully deoxygenated blood. We took the spectrum at 70% StO2, and the total hemoglobin concentration of 100 micromolar was used to calculate for the tissue absorption spectrum. Then the values at 405 nanometer and 630 nanometer were selected as the inputs for our simulations. The value of absorption coefficient at 405 nanometer was 34.1 inverse centimeter, which is approximately 100 times more absorbing than that in 630 nanometer. For tissue scattering properties, the values of reduced scattering coefficients can be predicted using mean theory as it follows the form of A lambda to the power of minus B. The value of B used in this study was 0.838, taken from Jack's paper for skin epidermis and dermis tissues. We use 10 inverse centimeter for the reduced scattering coefficient at 630 nanometer, which is the mean value we obtain at this wavelength in our other clinical studies. The μs prime value at 605 nanometer was determined to be 15.1 inverse centimeter from the theory. For the results, first we look at how light propagates in tissue for 405 nanometer on the left and light at 630 nanometer on the right. The colors on these figures are plotted on log E scale, and the log contour lines are indicated on each figure. Light penetration depth is defined as the depth when source power drops to 1 over E or around 37% of its initial value. From the plots, we can determine the penetration depths at each wavelength by looking at the -1 contour line along the central axis. From the simulation results, light penetration depth is approximately 0.9 mm at 405 nanometer and around 0.47 cm at 630 nanometer wavelength. It should be noted that these two figures are plotted in different scales on the x and y axis, millimeters on the left figure and centimeter on the right figure. Here is another figure with both 405 nanometer and 630 nanometer light overlaid on a single plot, just to show you how differently light penetrates at different wavelengths. In this figure, both the x and y axes are in centimeter. Generally, red light can penetrate much deeper into the tissue as it is less absorbed by the hemoglobin. Blue light, on the other hand, does not penetrate deep into tissue, less than a millimeter, due to the extremely high absorption by the blood. Next, we look at diffuse reflectance measurement with different source and detector separations at 405 nanometer. These figures are plotted on millimeter scales, and the colors are on log of 10 scale. The first figure on the top left shows the heat map of the light path taken by those light reaches the detector fiber placed at 1 mm away from the source fiber. About 99% of the detected light travel up to 0.8 mm into the tissue. When the detector fiber was moved 2 mm away from the source, on the top right, the amount of detected light dropped drastically, and only a very small amount of light was able to reach to the detector. This is completely understandable, as we have shown in the previous slide that light at 405 nanometer penetrate less than 1 mm into tissue. Further moving the detector away from source to 3 and 4 mm, the simulation shows hardly any light reaches to the detector. For diffuse reflectance at 630 nanometer, we see a very different light pattern. In general, light penetrates further inside the tissue. These figures have cm on both x and y axis, and the color are on log 10 scale. For source and detector of 1 mm on the top left, 99% of all light reaching to the detector travel up to 7.2 mm into the tissue. The probing depth decreases slightly when source detector separation increases. 7 mm when rho is at 2 mm, as shown in the figure on the top right. 6.9 mm depth when the rho is 3 mm, and 6.8 mm deep into the tissue when the detector is placed at 4 mm away from the source fiber. We further increase the distance between source and detector fibers, and at 5 mm separation, 99% of detected light travels up to 6.6 mm deep into the tissue. Around 6.4 mm deep at 6 mm separation, 6 mm deep at 7 mm separation, and approximately 5.6 mm deep into the tissue when detector is placed 8 mm away from the source fiber. We further increased the source and detector separation to 9 and 10 mm, and we were still able to see an appreciable amount of light reaching to the detector. For our simulation, the detector light travels up to 5.1 and 4.3 mm into the tissue at 9 and 10 mm source detector separations. From these simulations, we have learned that red light penetrates far inside of tissue and can reach to detector fiber placed up to 1 cm away from the source. This allows enough diffuse reflectance light signal for the extraction of useful optical properties information from the tissue. This result also allowed us to estimate the tissue sampling volume of each source detector pair, and this volume changes quite significantly from 1 mm to 10 mm separation. Overall, for 630 nanometer light, the tissue probing depth is between 4.3 mm to 7.2 mm deep into the tissue using our contact probe. For blue light in the highly absorbing region of the tissue spectrum, we don't expect light to travel far enough to even reach to detector placed at 2 mm away from the source. This indicates that our spectroscopy system is not able to pick up sufficient diffuse reflectance data at different source detector separations that are needed to extract for tissue optical properties in the blue region. Moving on from diffuse reflectance results, next we'll be examining the results for fluorescence simulation at 405 nanometer and 630 nanometer excitation and emission wavelength pair. In these results, as it involves stimulation of two photons at different wavelengths, to make it simpler for us to understand, these figures here show the heat map of the origin of fluorescence photons that reach to the detector fiber. Each figure is plotted with centimeter on both x and y axes. In these four figures, source and detector separations was increased from 1 to 4 mm. We see a very similar pattern on each of the plots, where there is a hot spot right at the contact between the source and tissue, regardless of where the detector fiber is located. Similarly, for each source detector separation, 99% of the detected fluorescence originated from within 1 mm tissue depth right below the source fiber. When we further separate the source detector to 5, 6, 7, and 8 mm, we observe the same pattern consistently where most of the fluorescence light that reach to the detector fibers are coming from the superficial tissue layer right underneath of the source fiber. The number of photons that were able to reach to the detector decreases significantly with increasing source detector separation. Beyond 4 mm separation, there isn't enough amount of fluorescence light reaching the detector. From these results, we found that the sampling depth of fluorescence mode of the contact probe remains unchanged for different source and detector separation. The detected fluorescence light was confined to a very small volume of tissue right at the source, due to limited light penetration depths of the blue excitation light. The fluorescence light generated was able to travel much longer distances to reach to the detector fiber placed several millimeters away from the source. However, the fluorescence intensity may degrade drastically when using larger S&D separation, without giving any other desired benefits. It is therefore more practical to use short SD separation to improve the signal-to-noise ratio of the detected fluorescence. As a conclusion, we demonstrated a quick tool using Monte Carlo modeling to perform numerical investigation on the effect of tissue optical properties and various probe design on light distribution in biological tissue. We simulated diffuse reflectance and fluorescence measurements with our contact probe design and examined the effects of varying source detector separation on the sampling depth. The results I presented in this study gave us insight to where the detector signal is originating from inside the tissue. It also provides us useful guidance on how the measurements should be taken using our contact probe setup to target certain tissue depths, and most importantly, it helped us understand better and interpret the measured tissue optical properties and photosensitizer concentration. Before I end my talk, I would like to take this opportunity to thank my advisor Timothy Zhu for his guidance and support, my fellow lab members Andrea, Whaley, Hongjing, and Andrew for their helps, and I would like to also acknowledge the funding support from the NIH. Finally, thank you all for your attention and have a good day. Thank you.